In this video, I want to show how to visualize a pathway and how to take the genes from that pathway and send them to Opentox-Garuda and get gene expression for the corresponding genes. So, because the pathway databases and the gadgets in Garuda are quite different, currently this does not work well for every pathway database. So you can see here we have many pathway-related gadgets, but they cannot all be used in the same way. And for the kind of analysis I want to do just now, in my experience, the IPAVS database works best. So I'm going to launch IPAVS. I double click. I click the red cross to close the help information. So this is a set of curated pathways that I can work with. And I'm going to select, for our purposes now, the calcium signaling pathway. And then I click the Discover button in the top right corner. And then I'm going to send the calcium signaling pathway to the species extractor. Now, the species extractor is actually a plugin for the cell designer. This cell designer is a tool that visualizes and edits pathways. So launching the species extractor will launch both the cell designer and the extractor. The point is just that the species extractor will prepare the pathway for gene extraction, and that's what we want later on. So I'm going to double click here. And then the cell designer launches. And it's going to take a little while to do the layouts. And there may be some layout warnings as we do this process, and then we just click OK. And now this has drawn the pathway for us. So here is the calcium signaling pathway in all its glory and complexity. Now, in the background, we have this little window called the species extractor. That's how I launched the cell designer, by starting the extractor. And it's going to extract all kinds of things from the pathway, not just genes, but also things that are modules or proteins or compounds and so on and so on. So it's a mixed bag of things. Nevertheless, we can use this. So I'm going to use this and click Discover and send all these things. And we're going to pretend it's a gene list, even though it's a mixed list. And I'm going to send it to the Opentox-Garuda data fetcher because I want to get the gene expression values for all these genes, insofar as they are genes. And here's the data fetcher, and it's going to ask me, the thing you just sent me is a sample list or a gene list. In this case, it's actually a gene list, so I select gene list and I click OK. So here's all those things received from the cell designer. Now, the genes in here are actually human gene symbols for the most part, such as these ones here. So actually, I want to work with human samples to make this work right. So to find the human samples, I go back to the Garuda dashboard, Opentox, I launch the sample finder. This is the Opentox-Garuda sample finder. I select human, and then it's got to be in vitro for human. And then I'm going to go with 24 hours, high dose, and acetaminophen. And then single dose and liver. I click launch. And we discover two samples, since there are only two replicates for in vitro. I'm going to select the sample IDs, and only the sample IDs, by clicking in this first column. Then I click data in the top right corner to do a data discovery. I click discover. And then I select the Opentox-Garuda data fetcher. I send those two samples to the data fetcher as well. And then it's going to ask me, I may have to switch to it manually. It's going to ask me, was this samples or genes? It was a sample list, so I click samples, OK. Now we have the two human samples here, and we have the gene list we've received from the pathway before, that's been remembered. So we're ready to launch, so I'm going to launch by clicking the launch button in the lower right corner. And now you see, for those things in that list that were genes, not everything was a gene, but some things were successfully identified as genes, and now we can see the expression value of those genes from that pathway for these in vitro samples from human. So that's how to go from the iPaths pathway database to visualizing the pathway and then retrieving that data and the gene expression from OpenTG gates. And this shows how gadgets in Garuda, even though they were not designed from the start to talk to each other, can talk to each other by communicating through the platform itself. So if you're interested, please try it out.